Obviously, Jessika needs no introduction for those of us who know her. But I still want to make sure everybody knows where she came from and how she got here. She was one of our undergrads and finished in 2016. I remember her very well from her undergraduate career. She is definitely one of the students that keeps us on our toes, which at the time I thought was really irritating, but now I've thanked her for that many times since then. She started the program in 2019, but in between, she went off to teach high school. So she's become quite proficient at teaching, has won the Outstanding Teaching Associate Award for graduate students twice, and has dealt with just about everything from dead clutches to that pesky little pandemic while she was executing this project. So she has persevered, to say the least. I think the amount of work she's done will definitely show as she goes through this presentation. I couldn't be prouder of her. So please welcome Jessika Smith.
Before we start, just double check on Zoom. Can you guys hear me? Can you give me a thumbs up? Yeah. Fantastic. Thank you. All right. Okay. Okay. All right, first off, I would just like to thank everybody who is here physically in person and virtually. Thank you so much for joining me on my thesis defense on seasonal dynamics and temperature effects on survival, hatching success, and morphological development in gremlin embryos. So, one of the reasons I love biology so much is in particular to the concept of climate change. I remember in the 7th grade when that concept came into mind that we need to protect the planet, and I just kind of latched onto that. When I started teaching, it really became my favorite subject to get into for my high schooler students, and I try to sprinkle it in there with my college students as well. So if you guys are my students and I pretended, right, I ask you to pair share, what do you think is happening due to climate change, particularly into coastal species? So you guys might sit around and talk, and I might get some of these as a response. So increased global temperatures, or more specifically for the ocean, right, ocean warming, ocean acidification, which could potentially lead to decreased oxygen in aquatic systems, which also deals with the stratification of the ocean. And that just means that the oxygen levels are changing throughout the ocean's depth. Or maybe you might give me a more simpler answer and say that there's rising sea levels. If you're more, I want something I can tangibly see and recognize. So maybe loss of key ecosystem services, such as mangroves, which is a living coastal protection, and seaside cliffs, which is a non-living coastal protection. But I think most people are familiar with, I think any biologist I can ask for sure, as far as like climate change, is the coral bleaching of coral reefs. But since I'm asking about specifically coastal species, and if I was probably talking to one of my high schooler students, they'd probably say, well, Miss Smith, the beaches are gonna get messed up, right? And so that's what I wanna focus on today. But before I get into my coastal species, I just want to recognize temperature, and temperature as a multifaceted factor. So in all of my time as an undergrad and all of my time as an instructor and continuing on with my education, temperature is in everything. So whether I was taking my cellular molecular biology class, talking about the fluidity of the membrane, in genetics, talking about genes being turned on due to temperatures, or maybe we talked about temperature as a selective pressure, a temperature preference in ecology, metabolic rate speeding up or slowing down in physiology. I never took microbiology, but I did take parasitology, and certain parasites thrive in warmer conditions. Photosynthesis for plants, pretty obvious, hopefully. And of course, animals. So my study was specifically on an animal, and so I'm gonna give you some examples of how temperatures have affected animals and their spawning. So for example, the Pacific mackerel in particular due to elevated warming temperatures, has not only delayed their spawning season, but it's also shifted more northward. And this makes sense, right? As the further we get up north in the globe, the cooler those waters will be. Another example is the European grayling, where they've actually advanced their spawning season 3 to 4 weeks earlier. So starting in 1970, it started approximately in the middle of March. And now over time, due to these warming temperatures, has now started earlier. But maybe you're not into fish, maybe you're into worms. So the navel shipworm is not a parasite, but it is particularly still a pesky problem. See, what they like to do is they like to feed on microorganisms inside of wood. And so they're a real havoc in shipyards. So with increasing temperatures, we actually see here in the gray that they've increased their timing of recruitment. And that just simply means that the larvae and the juveniles are reaching the populations. Not only are they reaching it earlier, but they've also extended it even further. So increasing populations of a pesky species. But I only care about coral reefs because that's the only, I know, okay, here you go. So sulfur leather corals, besides coral bleaching, let me give you a new fact. So for this one in particular, they have been disrupted their spawning time, varying at different depths. They prefer to be in shallower depths. They can go all the way to 45 meters. However, even though they can do that, it's also disrupting their synchronicity. So we have, in this example here, corals at less than 10 meters and 45 meters, they're not able to interact with each other. They're not able to continue spawning with each other. And that's obviously a big problem. So those are examples of how it affects animals. And it's, again, temperature multifaceted. So for coastal species, you could be a species that is specifically on the land or specifically in the ocean. Or you might be a species that is intertidal, dealing with both air temperatures and sea surface temperatures. So a lot more fluctuation. And it also depends where you are on the globe. So if anyone has never taken an astronomy class, right, or geography, because I hear they're getting rid of that. So the sunlight hits directly in the tropics. And as you go further north or further south, we start to get less sunlight, aka less temperature, less heat. So if you're a Pacific mackerel and you're shifting northward, right, that might not necessarily be a problem because the range here in the red is pretty much most of the globe. However, it can be a problem for species that do live in the polar zones, causing displacement towards them or competition at least. Or let's go back to our coral friend here, where they only are really found in the tropics. So currently, species that live in the tropics are already at their max tolerance, if you will, for physiological functions. And that means, okay, this temperature is kind of warm, but I'm still chilling, pun intended. I'm still regulating myself. However, with warming temperatures, this is going to cause more what's called, I'm pretty sure I remember from physio class, a physiological death or ecological death. Anybody who remembers that lab. And so it gets even more complicated when we start to add seasons. And so I brought up spawning species in particular because one, my species, I focus on their spawning. And so if they spawn in a particular season, right, temperature also plays an effect for that. So if they're spawning in spring or autumn, they're gonna have relatively equal amounts of sunlight versus if they're spawning in summer or winter, depending where they are in the hemisphere, they're gonna experience warmer or cooler. So if the behavior response, such as migrating, isn't enough, and we don't want species to die, right? Because we're biologists, we care about living things. Then maybe there's some sort of thermal tolerance. Maybe there's some sort of physiological adaptation that's going on. Are they able to tolerate warming temperatures? So I'm going to shift gears a bit toward my actual specimen, and it's the California grenion. But I'm going to just ask a quick question. Raise your hand if you've ever been to or heard of a grenion prior to today. Makes sense, all biologists. Okay, has anyone ever been to a grenion run? Okay, so I'm actually going to take you guys all on a grenion run right now. So here we go. So this is a typical grenion run, and what a grenion run basically consists of is grenions spawning on the beach. And so this happens between March and August. And the reason why is because the high tides in Southern California are at the max at this time. It's easy to distinguish the males and females. So the females are the ones with their tails entrenched in the sand and they're depositing their eggs. And the males are the ones surrounding those females, depositing their sperm. So these adult grunion utilize these tides to get themselves up onto the beach. Once they're done with their spawning behavior, they go ahead and use that same high tide to go back into the ocean. If anything, by the end of this presentation, I hope you go see a grunion run. It is so cool. So while those eggs are incubated in the sand, they'll incubate in the sand for about 14 days, but specifically for the next high tide, semi-lunar cycle. So that high tide is gonna come on in and essentially agitate those embryos. Those grunion embryos need that wave agitation. If they're not agitated, they will not hatch. And so that agitation, that wave hitting them is basically saying, it's time to go. So what happens is they release a chorionase, which is just an enzyme that helps them get out of their sac, the sac that's surrounding them. It's just called a chorion. So they use that, and not only is that wave telling them, hey, it's time to go, but they also use the wave to get themselves back on into the ocean. So if you're still interested, right, when do you want to go? You want to go around nighttime to see a grunion run, specifically when there's a full moon or a new moon. Now, a lunar cycle is approximately 30 days, but from full moon to new or new to full, it's about 14 days. So there is little known about the adult grunion other than the spawning behavior and the spawning season, but there's been a lot of research done on the embryos and rearing them and larvae, but I'm getting a little ahead of myself. So why embryos? Why grunion embryos? Well, because they're so really cool. They have such a resilience and determination. So what if they miss that tide? What do I mean by that? So if they miss their tide, they're actually able to extend their incubation. Again, they need that wave to hatch. They will not hatch unless they get that wave trigger. So in the laboratory setting, they've been actually able to extend their incubation about 3 tidal cycles, which is over a month, which is like a long time. So still interested? Okay, let me give you some more information. Okay, so where can you go to see these grunion run? So typically their historical distribution range is from southern-wise Punta Abrejos, Baja, Mexico, and has gone all the way up to Point Conception. However, in recent years, their range has actually extended more northward as far as Tamales Bay. And why do we think that is? Well, you probably are guessing correctly in your mind, it's temperature. So according to the California Climate Indicator report from 2018, they've actually indicated of all the regions in California, the southern coast has actually received the greatest temperature increase throughout average, max, and minimum. So with this in mind, with temperature just being involved in everything, with grunion being like the coolest species ever, and just overall, how do seasons, all of this in mind, this is what my aim was for my study, to investigate the influence of season on thermal tolerance in California grunion by specifically assessing them. Can they tolerate it? Can they survive? And can they hatch if they do survive? So how does temperature affect survival rate, or excuse me, incubation temperature affect their survival rate? How does it affect their hatching success? And how do seasons affect survival rate and hatching success? So for this study, I hypothesized that my summer month groups with increasing incubation temperatures would do better for survival rates and also hatching success. So just a little bit about the method. So where I had all my egg collections, I collected them in Topanga Bay, and that's in Los Angeles County or Malibu, California, for anyone in particular. I have Zuma Beach and Los Angeles Airport up here for a reason, that's for data to be shown later. So again, grunion runs observed at night. The following morning, collected those eggs. The seasons I went ahead and collected those embryos was for spring, and specifically March 2021, 2022, and April 2023. For summer, it was June 2021 and June 2022, And due to further statistical analysis, I actually had to categorize another season known as late season, and that was for July 2021 collections. So I collected them in the morning and I drove all the way back to Turlock and I sent them up in their little incubation chambers, and that would be considered 1 day post-fertilization. So just for pointing this out, DPF just means days post-fertilization. I reared them at 18, 22, 26, 30, 34, and 38 degrees Celsius, and each incubation temperature had 2 replicates known as replicate A and replicate B. I checked them for viability and development. So what does viable development look like? Well, just tracking them every day, right? If I see more organs forming over time, they're probably still good to go. But what about the ones that were not good to go? So basically any ones that got stuck in a particular stage or just overall discolored, deflation, and broken. So in the wild, right, if their chorion was broken, well, they're not gonna last very long if it's not time for that wave to come in. So these counts, which I did starting on day 5, every day up to day 14, I counted all the alive ones, all the dead ones, and did viable plus hatching, because I did hatching success trials, times 100%. So when I'm referencing survival rate, I'm specifically talking about on day 14, this is how many were alive out of the total that I had collected. And I'm gonna also reference this as first tidal cycle. I also wanted to do extended incubation. So I didn't count like 15, 16. I still try to keep them viable, or I still try to maintain them, but I did count for day 28 post-fertilization, and I'm gonna count that as second tidal cycle. So moving on to hatching success trials. So starting on day 10, every day up to day 14, I did hatching success trials. For each incubation temperature, 5 viable from replicate A and 5 from replicate B. So I went ahead and I put them in these petri dishes and agitated them with seawater. I agitated them for an hour and 5-minute intervals. And the justification for this is when we're out there on the beach, high tide is happening for about 2 hours, but I just wanted to peak that at an hour, and the 5 minutes would be the waves coming in and out. And that was kind of my justification for my protocol. Here they are swimming. They're so cool. I know I've showed a couple of people, if not in here, maybe on Zoom, in person underneath the microscope. So cool. All right, so hatching success was hatchlings over the number that were tested. And this was taken from Smider and Martin 2002. They had some slightly different definitions for it, so I just wanted to justify them for my study. So when I'm referencing hatching success, I'm specifically talking about just a number between 0 and 100. And the Smider and Martin, they were discussing 50% hatch in relation to this. So I just wanted to give respect to this, but just title it something slightly different, calling it successful ecological hatch. So if 50% or more hatched, it was considered a successful ecological hatch. And of course, hatching success trials were performed for embryos that made it to 28 days post-fertilization. So when I first started, I had not considered just morphological development as a concept. I was just like, wow, these things are cool. I'm just gonna take pictures of it like everybody does with a cell phone. Hey, this is cool, let me take a picture of it. So with that in mind, I actually ended up coming with some cool qualitative data. And so I categorized this as morphological development or morphological observations. And so when I was looking at them under the microscope, I was just curious as to what traits are shown when they reach what's called potential hatch readiness. So basically, like, what are the traits that are visible when they're ready to hatch? And so based off of this and Martin 2009, clear chorion. So see, this one's clear. This one is still a bit more orange. Dark melanophores, which are just the little black spots around them, a discernible red spleen, and observable heart and blood circulation. Again, so cool, right? And then and again, this was all done on my phone. So I was like very proud of myself when I got this to stand still for my phone. So you could see them twitching, you can see their eyes moving, and sometimes you can also see them doing a little loop-de-loop. It's really cool. So once they had all these characteristics and movement, I considered this, okay, they're potentially ready to hatch. As far as statistical analysis, for survival rates, I did a Kaplan-Meier for survival curves, but for significant differences, I did a log rank test. For hatching success, I did a two-way ANOVA, and if there was any significant differences for either survival rate or hatching success, I did a Holmes-Sadok post-hoc test. Okay, so we got through the intro, we got through the methods. Now let's get into the meat of it. What are the results? So how does incubation temperature affect survival rate, and how does spawning season affect survival rate? So based off of what I got from my data, seasons showed significant differences among incubation temperatures. So for those of us that are p-value happy, right, I got some really good p-values, okay? So for 18, 22, 26, and 30, with the exception down here for summer versus spring, that wasn't significantly different, but late summer was significantly different from spring and summer. And then when I went ahead and did it in the other way for incubation temperatures, there were significant differences among the seasons. So again, all our lovely p-values highlighted for spring, summer, and late summer. So here are my survival curves for spring, summer, and late summer. And it might be a little hard to see for those of us that are here in person, maybe not so much for those of us virtually, but just to give you an idea visually of what those curves were, since I did use Kaplan-Meier. And just seeing that 18 and 22 did relatively better in comparison to 26 and 30 degrees. But for those of us that want the numbers, I got you. Here's a lovely table for your convenience. All right, so if we just look here, 18, which is my coolest, and 38, which was my highest, we see it is decreasing in percentage over time. The only slightly differences I had was when we got to summer and late summer for 26 and 30, at least in relation to just their own categorization of 26 and 30. 34 and 38 degrees, that was basically like insta-death. They put them in the chambers and checked them and they were dead the following day. So even though I didn't use them for statistical analysis purposes, they, remember, 0 is also considered a result, so we add. All right, for second tidal cycle, only 18 and 22 had viability all the way up to 28 days, at least for mine. With the exception of spring 2021, I had a mishap, so I actually didn't count them in my statistical analysis, just specifically for 22 degrees Celsius. 26 and 30 had basically like negligible results. So, okay, those are my results, but what am I supposed to get out of this? Well, my hypothesis that summer spawn embryos would display higher survival rates with increased incubation temperatures wasn't supported for 18 and 22. So I'm gonna bring my lovely graphs again. And if I looked at 18 specifically, right, and just go across, so from spring to summer to late summer, I see that they had decreased over the seasons. And it's the same for temperature 22, right? Spring here, and they've decreased over the seasons. And just that simply just means that cooler temperatures and cooler seasons are just better for overall survival. Now, this has been shown in other species. So another climate change species that's been really investigated is the loggerhead sea turtles. And so they also spawn at particular, they have a seasonal spawning, and they experience high mortality during the warmer parts of the season. But I thermally tested mine on top of checking for seasons. So another example I have is the European squid. When they were thermally challenged, their winter eggs still did better in comparison to their summer eggs. As far as Brennan is concerned, the really cool, unique thing about them is that their embryos are able to develop on land, whereas the adults themselves, are in the ocean. Now, it's not necessarily uncommon for fish to spawn on the beach, but specifically for their embryos to incubate on land, inland, and stay and utilize it, right? The benefit of doing this is all the abundance of oxygen. That's mainly why they do it. So just grain of salt as far as like when it had to do comparison, I didn't really have a whole lot to compare. So for example, European squid, well, their embryos develop in the ocean and grunion develop on land, okay? Well, doesn't sea turtles' eggs develop in sand? Of course they do, but keep in mind, as far as they're very different. So as far as getting into the ocean, they need that wave. They need that wave trigger, and they also need the wave to physically get back into the ocean and remain there. Versus for sea turtles, they have lungs, but let's say we take lungs aside, they're still able to physically move with their flippers and don't need a wave to get back into the ocean. So moving on to my higher temperatures though, there was actually significant differences between seasons, not similar to 18 and 22. So for temperature 26, again, if I look at spring in particular, they had significantly better survival rate in summer, not late summer though. For temperature 30, they had better survival rate in summer and late summer. Now, even though my statistical test said that summer and spring for 30 degrees wasn't significantly different, it was still significantly different for late summer though. So those higher temperatures did better in those warmer seasons. And so it led me to think that if there are higher temperatures, incubation temperatures during those summer months, maybe there's some sort of adaptive response happening. So one idea that I thought could potentially be is transgenerational plasticity, and that's essentially whatever the parent has experienced is going to indicate some sort of response for the embryos, or their offspring, excuse me. So let's talk about thermal stress as one. So a cool adaptive benefit that I saw in one study is in coastal rainbow trout. And so both the parents were thermally challenged, and then they bred them, and then their embryos were thermally challenged as well. And so the offspring, the male offspring, actually had better survival rates when they were in the same conditions as their parents. However, with biology, whatever there's positive, there's going to be negatives as well. So this could also lead in the other direction, where in Mexican fried octopus, embryos thermally stressed from their mothers that were thermally stressed actually had lower survival rate. And if there was survival, they had smaller sizes, less yolk, and higher metabolic rates. But I wanted to point this out, transgenerational plasticity, again, leading to like the uniqueness of grunion, because adults spend their time, as far as what we know about the adults, are in the ocean, the embryos are on land. And so very different, technically, environments. However, regardless of that, we have sea surface temperatures and air temperatures have been increasing over, how far did I go? Pretty far back, over 50 or 70 years. So there is an indication for that. So I just thought it was a cool thing, potentially, maybe this is probably what's happening. I want to give an honorary mention to heat shock proteins. Part of the project had initially wanted to do this, but just due to a bunch of side step issues such as funding, timing, etc. Anybody who's done research, you get to do one, you think you're going to do one thing and you end up doing something else, and that's totally normal in research. But I did want to give an honorary mention to it as far as what could potentially be happening when it comes in relation to thermal tolerance. So in thermal tolerance studies, they check heat shock proteins, and for this reason, it's for improved heat resistance. They're seen in cell defense, morphological changes in development. And what they essentially do is they, for the most part, help in refolding misfolded peptide proteins. So heat shock proteins are conserved, but they can also be induced when the cells are stressed out. And they've also been seen as far as an upregulation in embryos, not just in our lovely friend, the loggerhead sea turtle, but fish species as well. But what's probably most likely happening is metabolic rate adjustment. And that's just the organism's ability to regulate its metabolic rate. Because as ectotherms, they don't necessarily get to control the temperature that they're in. Not like us ectotherms, right? We have a core temperature our body's able to do. So in order for looking at metabolic rates in research, we look at metabolic biomarkers. And so one example for developing big fin reef squid as a response to heat wave treatments, there was an uptick in, oh wow, look at that heat shock protein 70 in thermal tolerance, but also other biomarkers such as cytochrome oxidase, which is related to aerobic metabolism, and superoxide dimutase, which is an antioxidant molecule. Of course, there are other biomarkers we could look at, such as lactate dehydrogenase, which is an anaerobic metabolism, but this was just the study that I had. So with all that in mind, this is what I think happened. But there was another study, the Smider and Martin study, that also did survival rate and hatching success. So I just wanted to compare our studies like any good scientist. So the Smider and Martin had 80% for all their incubation temperatures, 80% survival rate, whereas my only one that was over 80% was for 18 degrees. And what I think potentially probably why we had such differences is a few things. So one was our incubation setup. So the Smider and Martin had their embryos, so they're in the containers, right? And in the Smider and Martin, they did theirs in sand. And it was moistened because they need that moisture so they don't desiccate with deionized water. Again, not really fluctuating any solidity. Excuse me. For my study, I did 2 paper towels. They were moistened with seawater to, indicate sandy environment, if you will. But I think that was probably one of the reasons why I might have had more death in comparison to this study. Incubation temperatures was obvious, but I didn't really, I don't think that was necessarily it because our ranges were particularly similar with Smider and Martin veering on the cooler side versus my temperature range is a little bit more warmer. I also just wanted to double check our collection dates. And so obviously the difference between this study and my study as like a whole concept for the research, my added component was looking at seasons. And so I have a bunch of different data for that. However, when I went to go ahead and just compare our Junes, right? Because in the Smider and Martin, they just collected in June. So I went ahead and I wanted to compare our June. And so the Smider and Martin collected in Malibu Beach. I collected mine at Topanga Beach. Los Angeles Airport is where I got my air temperature data. So everybody is relatively close. So no like latitudinal as a factor. So I went ahead and I got the data all the way back to 1950 up to 2022. And I don't know if you can see in the blue, that's the Smider and Martin. And then the 2 red were my study. So I checked June average temperature and it's cooler, but only in comparison to 2022. But I wanted to be extra thorough. So remember, when do we do Brennan runs? We do them at nighttime. And then when do we collect? We collect them early in the morning. And so those temperatures are relatively cool. So it just made more sense to use June minimum temperature as far as a comparison, because minimum temperatures reflect the overnight low temperatures anyway. And so once I did that, I noticed that, that the Smider and Martin had cooler temperatures in comparison to my other 2 for 2021. One last thing that I considered maybe was potentially causing such differences too is the timing of our setup. So the Smider and Martin, their laboratory is at Pepperdine University. So here's Malibu Beach, again, Topanga Beach. It's about not that far, 15 minutes. Even if you consider LA traffic, you could still probably get there in less than 30 minutes. Versus for me, I had to go through LA traffic and go through the Grapevine and go all the way back to Turlock, which took me approximately 5.5 to 6 hours. So they were in my car in a cooler. And that is a critical stage for any embryonic development, is those first hours or days, depending on how long their development is. So those are things that I considered why we had such differences. So speaking of timing, I want to shift gears into my next set of results, which is my morphological observations. Now, I know I'm skipping hatching success. I promise I will get to it. But my morphological observations has to do with the timing. And that's what I found when I was looking at them and comparing my pictures of my different temperatures and seasons. So overall, I just noticed increasing temperatures had accelerated the development of morphological traits. And remember, the morphological traits that I was interested in is what do they look like when they're probably ready to hatch? So I was just in my room comparing my pictures, and this gave me like the realization, hey, I see something going on here. So I just picked a random day, so day 4 and summer group. At day 18, this is what they look like. So they're orange oil droplet. I don't know if you can see little eye spots forming. They are slightly forming. 22, again, same day 4. We see distinctive eye spots, those little black dots, those are their eye spots. 26, they have more distinct eye spots. We can see the red spleen. We should be able to see heart rate, blood flow, and it's becoming less orange. And for temperature 30, again, same day, it's less orange. Everything is more distinct. There should be movement happening. You can't really show movement on the picture. You get the idea, right? All the same day, but where they are developing is very different. And so I looked at the pictures that I had for all my seasons and all my temperatures. And again, these numbers reflect the traits as far as for potentially being ready to hatch. So that red spleen, the melanophores, the distinctive eyes, heartbeat, or noticeable heart, etc. And so for temperature 18 across all seasons, they didn't get to this point until 11 days post-fertilization. For 22, 7 to 8 days, 26, 5 to 6 days, and 34 to 5 days. Now, I don't really have any statistical analysis for this, because again, this was just something that I just thought was really just cool to point out. Let's just take it as our qualitative data, if you will. So, what the heck does that mean? Well, increasing temperatures accelerated embryogenesis. And I can't really necessarily outright make that claim without any sort of metabolic data, but I can say metabolic rates do accelerate with increased temperatures, and this has been seen in other studies. So for metabolic rates, they either check oxygen consumption rates or a specific metabolic biomarker. So in this study, as temperatures increase, we see oxygen consumption increasing over time. And again, this is an embryo study. In another study done for zebrafish, now granted, these zebrafish were adult zebrafish, however, they were reared when they were in the embryos at different temperatures. So zebrafish, according to this study, their ambient temperature is 27 degrees Celsius, and so they were had warming treatment for 32 as far as comparison. And so this actually had a persistent effect into their adulthood, showing that the higher temperatures actually, for the adults, had higher lactate production. So if we're speeding up our metabolic rates because we're increasing our temperatures, well, then we're gonna be requesting more metabolic demand. And so what does that mean? We need more energy. And if you're an embryo, you can't exactly get out of your egg and go eat something. What are you eating? What are you getting your energy from? You're getting it from your yolk. And so increased metabolic demands actually increase the usage of yolk reserves. And this has been seen in plenty of studies, but here's just one study that I do have. So in relation to the yolk area to the egg, we see as temperatures increase, the area of the yolk decreases over time and with increasing temperatures. So this is probably most likely explains why higher temperatures had a lower survival rate in comparison to lower temperatures. It's hot, and they're dealing with, okay, I'm trying to keep up with this heat. My metabolism's trying to deal with it, and they just probably most likely ran out of yolk. So I wanted to show this graph really quickly because there has been oxygen consumption study done on embryos. And so this study by Darkin, they incubated them at 18 degrees Celsius. And what they found is between developing days 1 through 8, you see a sharp incline up to metabolic rates. And by about day 8, it starts to level off. And so even though it's leveling off, that metabolic rate is still relatively high. And so in another study in relation to this, I wanted to point out is Martin 2009. And well, hatching competence is met at 7 days post-fertilization, but that's in relation to 25 degrees Celsius. So I just want to sneak peek this for a second. Now I want to talk about my hatching success. So how did incubation temperatures and seasons affect hatching success? Well, strangely enough, seasons showed no significant differences for hatching success. Incubation temperatures did. So I wanted to look further into that. And so I went ahead and did days post-fertilization against incubation temperature. And there was a significant interaction between those two. So the data for that, I have, if we look here at 18, which is represented in black, we see that it is increasing over time. For 22 and 26, it's relatively the same throughout all the hatching success trials. And for temperature 30, we see a slight decrease over time, starting off strong with that first day, but starting to decrease as we get further into the development. As far as overall, right, what was it considered an ecological success? So for all the temperatures except for temperature 18 had a successful, an ecological successful hatch for first tidal cycle. When I went ahead and tested for hatching success for second tidal cycle, 18 finally shined. It finally met threshold for over 50%. 22 also was still over 50%, slightly decreased from first tidal cycle, but still relatively high. And 26 had decreased, and 30, I didn't have any that lived that long to do any hatching success. So what does this mean? What do we get from these results? Well, temperature is the significant key factor in comparison to season. Season didn't really have any effect on the hatching outcomes. And again, this has been seen in other ectotherms. So if it's too cool, you might stop your development overall, hence you never get to actually hatch because you haven't got to the stage of being able to hatch. Or, you know, if the temperatures are too high, again, going back to the metabolism thing that I talked about earlier, it's decreasing in hatching success. Whether it's higher temperatures or cooler temperatures, anything outside of the norm. And just depending on how warm or how cold it is, it could just lead to overall a not successful ecological hatch. Again, just depending on what's going on. So hatching success in relation to temperatures and the rate of embryogenesis. Again, and this is why I wanted to talk about morphological development first, because it is my bridge between my two data sets. So increased temperatures were associated with accelerated developmental rates. And this has also been seen in other Grenion studies. But again, I don't have that metabolic data to back it up. So I have all these other studies to help me back up that claim. All right, so potentially increased metabolic demands. I want to focus back on our friend, the European eel. As in this study, it clearly defines what I think is happening to my Grenion. So increasing temperatures accelerates the developmental rate. So that just means it takes less time to get a specific point. And in this case, the study was looking how long does it need to develop before it's able to hatch. So in their study, they compared, and their highest temperature had the least successful hatch in comparison to the other temperatures. However, their yolk utilization, as temperatures increase, the yolk utilization increases. However, if it's too stressful or too hot, even though they might be demanding more of the yolk, we might run out, or we just might be inefficient at it, and that was demonstrated at higher temperatures. Again, everything with a grain of salt. This species develops theirs in the salt water, and then they migrate all the way back to fresh water. But again, I feel like the concept still applies. So successful hatching depends not only on temperature, but it depends on the timing. Are they ready to hatch? Are they at the critical stage for them to hatch? So based off of my ANOVA, yeah, there is a relationship between the two, and that justifies that. If I just look specifically at this graph in particular, certain days for 18 and 30 had to increasing or decreasing versus temperature 22 and 26 seem to be the preferable incubation temperature. Now, again, the Snyder and Martin also did hatching success, and I wanted to be good and compare the two. So our studies were actually, getting a little ahead of myself. Our studies were actually relatively similar. So in their first tidal cycle, again, they did 15, 20, and 25. Their lowest temperatures did not meet, or mine and theirs did not reach successful ecological hatch, remaining below the 50% line, versus our higher temperatures did meet that threshold. For second tidal cycle, our lowest temperatures finally did meet that threshold. Our slight differences was 25, still met the threshold, but for mine 26. So what the heck is going on? We're just tying it all together, okay? So whether you are delaying your development because your temperatures are cool, or maybe your development is happening a lot faster, right? When we get to here, this is all that matters as far as, okay, we are potentially hatch ready, but are we gonna run out of yolk reserves, or am I gonna get to develop in time in order to reach my hatching potential? So whether you rely on a trigger or whether you rely on a different factor for hatching, these things matter. So tying everything together, what I learned, and hopefully I didn't lose anybody, let's get back on track and just summarize everything. Temperature just plays a pivotal role in shaping the outcomes of Grenion embryo development, and it has significant implications in their survival and their overall hatching. Survival rate, spring spawn embryos incubated at 18 and 22 displayed higher survival rates in comparison to summer and late summer. And this just highlighted the role that seasonal temperatures do play a part in embryo viability. For Grenions incubated at 26 and 30, they had a higher survival rate during summer and late summer respectively in comparison to spring. And this raises potential questions on, is there a thermal tolerance? Is there some sort of mechanism that allows them to do that? Hatching success. Hatching success in Grenion embryos was significantly influenced by incubation temperatures, but not necessarily seasons. Optimal hatching based from this study was 22 and 26. This just underlines the importance of thermal control during embryonic development. It also brings attention to the physiological adaptations, that data that we need in order to relate the two, which brings me to my morphological observations. So temperature accelerated the development of morphological traits for those who are potentially ready to hatch. And again, just underlines that we need a baseline for metabolic responses, not just for one particular temperature, but in different types of stressful conditions, whether that's increasing temperatures or other types of stressors. And thus bridges the gap between survival rate and hatching success, right? It's great if you have high hatching success, but if you're not able to make it that long, that's kind of an issue. So just a couple of things that I hope that this research can help with. Obviously, the big one is climate change. That's the reason I'm doing this, because climate change is important to me. Grenion are actually considered an indicator species for climate change on beaches. And so, as earlier I stated, they are moving their range a bit more northward. However, their runs have been becoming increasingly low. So when I first heard of Grenion runs, and when I first saw them on a YouTube video, I saw that there was like fish scattered all over the beach, like so much that you couldn't walk. So I was so stoked. I was like, oh my God, that's gonna be so cool. And so I get out there and I'm like, okay, I see fish, but I can still walk around them and not step on them. And that is becoming more and more normal. It's becoming decreasing as far as seeing the whole beach scattered with entire fish where you can't walk. So there are current climate change research on Grenion, so like ocean acidification, which in those conditions increase mortality, and warming ocean increased feeding and thus respiration. But these studies have been done on Grenion larvae, not specifically embryos. And the reason I want to point out embryos as being super duper important, because they are the indicator species, but also because they develop in the sand. And so I hope that this research also helps with informing policy on conservation efforts for California Grenion, because they are highly influenced by human activities such as urbanization causing erosion on their beaches, which is what they need in order to develop, and of course due to recreational fishing. So for July, my late summer, I only have one data set for that, and that's because I actually did go out there twice in 2022 in July. But due to the amount of people, because it's an open season for Grunion in July, I wasn't able to get anything. The Grunion weren't able to do anything. They were there, but they weren't able to do their spontaneous behavior because there were so many people on the beach. So with all this in mind, please remember Grunion are friends. They're super important. And thank you. I want to say thank you to my family and friends who've been there supportive of me. I want to thank Alvin in particular for coming out with me on my field to help me do field work, and Zach as well. I want to thank my Edison High School colleagues because without them, I don't feel like I would have felt brave enough to leave my comfy position and pursue graduate school. I want to thank my fellow graduate cohort. I know some of them are watching right now, and oh my God, we finally did it. I'm finally going the finish line, guys. I'm finally there with you. It's been a wild ride, and I just appreciate you guys so much. I want to thank the Stanislaus Biology Department for my entire experience here. As an undergrad, as a credentialed student, as a graduate student, I have been so blessed with professors that I feel like gave a shit, excuse my language, I couldn't help it, about my academic career and my aspirations. None of this would have been possible without that support, that emotional, yes, you can do it support. Of course, I want to thank my committee members. I want to thank Dr. Karen Martin, who is watching virtually. Hi, Karen. I appreciate you so much as a colleague, as a researcher, as someone I admire, as someone who is doing out there the climate change work. I appreciate you so much. Thank you for all the knowledge. Thank you for letting me stay at your house. Thank you for giving me such advice and just gearing me towards a species that is super important to the environment. I want to thank Sarah right up here. There have been back and forth as far as like, do I want to do research or not? She was actually the first person to let me do research with her, specifically for bioeducation, but she let me dip my toes into it and just helped me encourage myself to just, why not? The worst that's gonna happen is a no. And so I just appreciate you and thank you for all your emotional support and just kind of guiding me in that starting point. And of course, I want to thank Brian, who has been nothing but supportive and understanding. We went through COVID, we went through some other BS that we don't need to get into, but you know, I appreciate you so much as a colleague, it's weird to call you a colleague, as a student, as a colleague, as a mentor, as an advisor. I feel so successful. Thank you very, very much. And that's it.
Time for any non-committee member questions, including those out there in Zoom land. So no Karen, no Sarah, no me. Catch your breath for a minute.
Yes.
So during your hatching experiments, did you monitor other abiotic parameters during the time period when you were making observations, like oxygen, pH, salinity, and so on?
I wasn't necessarily tracking any of the other abiotic factors. I just tried to continue to do my hatching success trials in the same location every time with the same petri dishes, with the same seawater, held at the same temperature. So I wasn't able to track necessarily salinity, but as far as I made sure to use the same salinity of seawater when I did my hatching success trials. Yes.
So during hatching, what variance of temperature did you have when you used the water? Was it like plus or minus 1 degree, or were you very diligent about making sure it was 18, 22, 26?
So that was actually a discussion up for debate during my thesis proposal as far as determining that temperature fluctuation. So I tried for the most part to, when I had the seawater, I had it in ice and I monitored it just like every so often, just kept looking at it, and then I took it off the ice to make sure it stayed at 10 degrees. If it started to go up, I'd put it back on the ice. So I tried to be as diligent as I could. I don't actually have the error numbers for that. It was just more of like, this is what I'm seeing right now, because I'm monitoring the temperature, but every 5 minutes I'm also agitating, I'm shaking them, right? I'm looking, oh my God, did they hatch, you know? So, but that is a good question. Thank you. Yes.
I'm kind of struck by how the survival rates are different depending on what season the eggs were collected, right?
Mm-hmm.
And I'm wondering what your thoughts are in terms of why do you think the fish are even laying their eggs later in the season if the success rates start to drop? Does that make sense?
I think so. So for peak spawning season is April, May, and June. And so actually recently, it used to just be as far as like, if we're talking about recreational hunting, April and May were closed seasons. They just recently now included June as a closed season, which I think the first time I went out there, June was still technically an open season. And again, those people up on the beach caused such a havoc for them, because even like going out there with your flashlight, right? Because they're there out in the ocean. They send one to come up, and then based off of that, right, like, are we gonna go or not? So there's just the disturbance of people up on the beach at nighttime could disrupt that. I hope I answered your question. Okay.
Right.
Yes.
I don't know about grunions, so like, do they spawn once a year, or are there different? Like, could the same ones who spawn in June also be the same ones spawning?
Right.
Again?
So if they're able to spawn multiple times throughout the year, so like in the early seasons of like March, it's going to be the grunion that had developed from the prior season. Of course, there are like 2 to 5 year old grunions out there. Based off of what I read from Game and Wildlife, it's most likely the 2-year-old grunions out there spawning for the entire season. As we get later into the season, grunion that I guess had hatched like earlier in the season could potentially also spawn up on the beach later in the season.
Just an interest question. People are out there fishing for grunions, or are they fishing for other things?
They're fishing for grunions. So when I was out there, they would have like buckets or nets, which you're not supposed to. There's just like a limit on which grunion you can take. And of course, I'm out there yelling at them like a crazy person, at least to not take the females, right? And so you can eat them, but people usually use them for bait for bigger fishes.
Okay.
Yeah.
Thank you.
Mm-hmm. I think I saw another hand. Yes.
Do you recall the concept of survivorship curves from ecology, the type 1, type 2, type 3 survivorship curves?
Uh-huh.
And this typically means that we follow a cohort of individuals, from birth to their eventual death, and we can sort of chart out 3 sort of major strategies to this. So I was kind of struck by one of the slides that you showed based on the season of run and the hatch success of those different runs at these different temperatures. And I was totally seeing like type 1 survivorship curve for the March group, type 3 for the June and July group. There's this big drop off straight away in survivorship. And are there data out there for the species about their actual natural survivorship curve, what that looks like? Do you know any of that?
As far as when I looked at survivorship for grunion, is the studies that I found, they usually collected on a specific month, not necessarily like a long over period, like from the March to August. There was one study that did, but it was quite old, and they were specifically also looking at like sand temperature under there. So it was a little bit more difficult to read, but as far as like all the entire season, I am actually not sure. As far as like the stuff that I've read, it's just been a particular month where they collected.
Anybody else? All right, one more time.